Good afternoon, students. Myself, Mrs. Ashwini Kadbhane from Department of Microbiology, S.V.K.T College, Devlali Camp. Today we are going to see the SYBSc semester number 4, that is the genetics. In that, today's chapter name is Understanding DNA, and our today's session is Evidences of DNA as a nucleic acid. The experiment of DNA as a genetic material were carried out by various scientists. The two experiments we are going to learn in our today's session. So, here we before starting our today's session, we are going to recap what we have seen earlier. In our previous lecture, we have seen the search of genetic material, what were the initial discoveries and the history of the DNA for this DNA structure. We have seen the first experiment, that is the Griffith experiment. Griffith proved that the transformation experiment, in which he proved that there is something which is going to be transferred from one progeny to its next generations. So that experiment was seen. He chose the microorganisms, and the two colonies, that is the smooth and the rough colonies, were selected for that, and he did the experiment which was seen in our previous lecture. We have seen its conclusion also. The next experiment was seen by us, that is the Avery-McLeod and McCarty experiment, which were the further proceedings of the Griffith experiment. They carry out his experiment and they proved that it is the DNA molecule which was transferred from one progeny to the next progeny, and its conclusion. So our today's session agenda is to see the next experiment, which was given by the Hershey and Chase, and which was a bacteriophage experiment. Okay, Avery-McLeod and McCarty, they explain as not in only bacteria but viruses, they also have the genetic material. And the bacteriophage means a virus which infect the E. coli. So in that, how did it the experiment and what was its conclusion, we are going to see in today's lecture. Guaranus-Carmen tobacco mosaic virus experiment, that is the TMV experiment, which was concluded, which was done in 1956, and its conclusion. Franklin Corhant and the Singer in 1957, their experiment and their conclusion. What is known at this point in the time? First, we are going to recall that what were the findings till now. In 1848, chromosomes were discovered, exchanged during the mitosis, and it was the theory was given by the Wilhelm Convenster in cell nuclei resolved into small rod-like bodies during the mitosis. After that, Friedrich Mischer, he told us the nucleon, that is the term nucleon, which contained the phosphorus-rich molecule, and the DNA was isolated from the cell. That was done in 18th century. In early 20th century, Walter Stutton hypothesis that chromosomes are hereditary units. Chromosomes carry the genes, basic units of the heredity. Genes are generally linear on the chromosomes. Chromosomes are made both from the proteins and the DNA, which is known as the carrier information. This was told by the Walter scientist. Then in 1928, Frederick Griffith, he took an experiment by using a pathogenic strain, that is the Streptococcus pneumonia, and the bacterial transformation, that is the transfer of genetic material, was explained. But till the date, he was not knowing that is it a protein or is it a DNA which is going to be transferred from one progeny to the next progeny. So in 1948, his findings were being concluded by the Oswald Avery, Colin McLeod and McLean McCarty, who determined that it is the DNA, not the protein, which is transformed from one progeny to the next progeny. He used the various enzymes like DNase, RNase, proteinase to break down the cell and to do the experiment. And later on in 1952, Hershey Chase, he did the experiment which proved that in bacteriophages also, the DNA is the only genetic material which is passed from one progeny to its next progeny. For transformation, there is some criteria for the genetic material. What are the criteria we are going to see in this slide? Information, it must have the information necessary to make an entire organism. Transmission must be from one parent to its offspring, it should be transmitted. replication should be there accurately. Progeny cells have the same genetic information as the parental cell. So, replication is very important part of a genetic material, and it should be have a variation. It must be for capable of changes. Without change, organism would be incapable for its variation, and adaptation and evolution could not occur. So, identification of the DNA as a genetic material. Before that, any genetic material which is passed from its one generation to its next generation should have these types of the criteria. So, now we are going towards the Hershey and Chase experiment. DNA, he explained the DNA as genetic material in bacteriophages, that is in viruses also. Even though research discovered that the factor responsible for its inheritance of trait comes from within the organism. So, they failed to identify the hereditary material. The chromosomal components were isolated, but the material which is responsible for inheritance still remain unaware. So, Griffith experiment was the stepping stone for discovery of genetic material. So, it took a long time for acceptance of the DNA as a genetic material. Let's go through the discovery of DNA as a genetic material. So, scientists Alfred Hershey and Maratha Chase, while DNA had been known to the biologists since 1869, many scientists still assumed till the time that it is a protein or it is a DNA which is carrier for information which is inheritance, because DNA appeared to be inert molecule since it is located in the nucleus. Its roles to be considered to be as a phosphorus storage, because we have seen in a previous slide that one scientist who told us that the DNA molecule consists of the phosphorus components. So, till that time they were thinking that as DNA may be a storage for a phosphorus, and protein it can be a carrier or it can be a genetic material. So, in Hershey and Chase showed that bacteriophages, which are composed of DNA and protein, they infect the bacteria. Their DNA enters in the host bacterial cell, most of their protein does not. See, in bacteriophage, it is the virus which is going to infect the E. coli. So, in that it will incorporate its genetic material, not the protein code, which was proven by the Hershey Chase. And after his experiment, it was totally agreed that it is the DNA, not a protein, which is a genetic material of each and every cell. Hershey and Chase subsequent discoveries all serve to prove that DNA is the only hereditary molecule material present in each and every cell. Hershey shared the 1969 Nobel Prize in Physiology of Medicines with Max Delbruck and Salvador Luria for their discoveries concerning the genetic material. In 1952, Alfred Hershey and Maratha Chase took an effort to find the genetic material in organism. Their experiment led to an unequal proof to their DNA as a genetic material. Bacteriophages, that is the viruses that affect the bacteria, were the key element of their experiment. So, you can see in the picture that they are the Hershey and Chase with who did the experiment on the bacteriophages. The viruses, now all we know that the viruses, they don't have their own mechanism for the reproduction purpose. They only have the genetic material. So, for the reproduction purpose, there is the need for them to in search of a host, and a particular virus will infect a particular host only. All the virus cannot infect the E. coli. It is the T4 virus, T4, which can infect the E. coli. So, it is called as the bacteriophage. The virus, it doesn't have their own mechanism of reproduction, but they depend on the host for the same. Once they attach to the host, their genetic material is transferred to the host. Here in case of bacteriophages, bacteria are their host, so they are called as bacteriophages. They infect bacteria, are manipulated by the bacteriophage such that bacterial cell starts to replicate the viral genetic material. What they do? They incorporate, they attach on the bacterial cell membrane. They lyse their cell membrane and try to incorporate their genetic material into the bacterial cell. Now what happened when there is incorporation and penetration of their genetic material? At that time they make a condition in the bacterial cell, so that bacterial cell thinks that the viral genome is its own genome, and due to this thinking it tried to reproduce and replicate the viral genome, and because of that many viral genome they are being produced, and the because of the production of viral genome there is the production of the capsid, which is the outer protein coat of a bacteriophage, and then there is a lysis of the cell and all the phages they comes out. So this is the lytic and the lysis cycle which is followed by the bacteriophage, but it is done in the experiment of Hershey Chase. Hershey Chase conducted an experiment to discover whether it was protein or DNA that acted as a genetic material which entered in the bacterial cell. So before studying about their experiment, we are going to see how the bacteriophage look. You can see in the diagram bacteriophage structure, which is having a head, a tail and the tail fibers. In the head is consisted of a protein coat. There are various proteins present on the head. Inside the head there is a DNA, which is a genetic material of that phage. There are some internal protein. There are some integrated and the peripheral protein which is present on the bacteriophage structure. After that there is a neck, there is the tail sheath, and including that tail sheath there are the tail fibers, end plate, pins which are used to attack a host and for the incorporation of the genetic material. Now we are moving towards their experiment. They grew one sample. Now as we know that DNA, they were also knowing that a DNA is composed of a phosphorus. We know that the structure of DNA is composed of the ribose and the phosphorus backbone and the nitrogen bases. They are present inside the DNA molecule which having the hydrogen bond between them. So the phosphorus and sugar molecules, they are the backbone of the DNA. Till that time, Hershey and Chase were knowing that phosphorus containing molecule is the DNA. So what they did, they took the two molecules. One it is the sulfur containing and one it is the phosphorus containing. Both were radioactive molecules. Because of the radioactivity, they could know that which molecule have been transferred or transported towards in the bacterial cell. So they grew one sample with the 32 phosphorus, which is a radioactive isotope of phosphorus, and the other sample were grown in 35 sulfur, which is the radioactive isotope of the sulfur, for many generations. The protein coat has sulfur, so no phosphorus, while the DNA material has phosphorus but no sulfur. So they selected the sulfur and the phosphorus molecules. We know that a protein molecule may contain a sulfur, but a DNA molecule doesn't have a sulfur molecule in that. So these two molecules were selected by the Hershey and Chase. So two samples were labeled and two different radioactive isotopes were selected. So what they performed in 1952 using bacteriophage, a type of virus that have a very simple structure, an outer core and the inner component. The phage are made up of equal parts of protein and the DNA molecules. It was known that phage infect by the anchoring the outer cell of the cell surface, and they deposit their inner component to the cells infecting them. Scientists were interested in finding out whether it was a protein component or the DNA component that is deposited inside the cell. So this experiment was carried to find what is the component which is being incorporated by the phages in the cell. By incorporation, radio label either in the protein or the DNA that infect the phage. They determined that DNA was isolated, introduced to the infected bacteria, causing the proliferation of the new phages. So experiment was carried out. What was the experiment? E. coli was selected as the bacteria because bacteriophage T2 was selected. So T2 bacteriophage infected the E. coli. In one part of experiment, T2 proteins were labeled with the 35 sulfur, and the other part T2 phosphorus molecules DNA was labeled with P32, that is radioactive phosphorus. Then each group of labeled virus were mixed and separated with the E. coli host. Both the samples were kept separated and the E. coli host was provided to them, so the infection would be occur. After a short period of time, phages attach was disrupted with the kitchen blender and the location of the label was determined. The 35S labeled protein was found outside the infected cell, while the 32P labeled was inside the E. coli, indicating that DNA carried the information what needed for the viral infection. This provided the additional support for the idea of the genetic inheritance in the bacteriophages. So, in the diagram you can see the two samples were taken. In first, a sulfur labeled protein capsule, and in second, a phosphorus labeled DNA, which was shown in green color. So, the red one, it is the coat of the protein, it is the coat of the phages, which is in proteinic in nature, were radioactive isolabile by the sulfur S35 and the phosphorus S34 were radioactive isolabile. A host cell, it was provided to the virus, and then the infection occurred. In the first step, you can see the infection occurred, and in both stages the DNA was incorporated. Now there was the blending by the kitchen utensil, and due to the blending you can see the bacteriophage DNA was mixed with the host DNA. After centrifugation, no sulfur in the cells were seen in the host cell, but after centrifugation phosphorus in the host cell were seen. So, this means that the sulfur containing molecules were not passed by the phages, but the phosphorus containing molecule were passed from the phages into the host cell. Again you can see in the diagram by using this experiment, again we can see in the diagram there is the batch 1 and batch 2. One, it is the radioactive sulfur, that is S35, and second is the radioactive phosphorus, which is the P32. Both the host cell were mixed and radioactively labeled. Now you can see in the first batch the radioactive labeled sulfur bacteriophages were incorporated in the bacterial cell, and after their infection they were centrifuged or they were blended. Okay, so they were blended, the empty protein shell were found and phage DNA was incorporated in the cell. Now they did the centrifugation. Due to the centrifugation, the pellet and the supernatant, they were separated from each other after centrifugation, and in the pellet the bacterial cell were found. In that there were no radioactive sulfur found. But in the second batch, when there is the infection, after the blending, after the centrifugation, in the pellet they got the radioactive phages or radioactive DNA or radioactive phosphorus, which means that the sulfur molecule has not been transferred, but the phosphorus molecule is being transferred, which is the DNA, which is a DNA molecule, and this experiment proved that DNA is the only genetic material which is passed from one progeny to its next progeny. Again you can see in this diagram, same diagram I have been showing to you. Again you can see a T2 phage which is infected in E. coli and grown in phosphorus containing medium. The cell are been lysed, it is been blended, it is been centrifuged. You can see progeny phage with phosphorus labeled DNA. In second step you can see the infected E. coli grow in S containing 35 containing medium, and the blending and centrifugation is being done, and the progeny phages with S labeled protein were not found. So, experimental showed that DNA is the genetic material in T2 phage. So, radioactivity recovered in the host and passed on the phage for the progeny, and in protein radioactive were recovered in the phage ghost but not passed on the progeny. Ghost means the outer covering of the phage without the genetic material. It is called as the ghost. So, here the ghosts were found without a genetic material. So, this proved that the DNA is only genetic material which is passed from one progeny to its next progeny. This experiment was carried out by the Hershey and Chase, and for this experiment they got a Nobel Prize also. So here also same diagram, T2 phages, E. coli, blender and centrifuge are used, and you can see the radioactive recovered in phage ghost, and next one you can see radioactive recovered in the bacteria, which told us that their DNA is only genetic material. So their observation in the first sample were phosphorus 32 was used, the bacterial solution showed radioactivity, whereas the supernatant barely had the no radioactivity because in supernatant there were the proteins. In sample where 35S were used, the bacterial solution didn't show any radioactivity, but supernatant did because in supernatant there were the proteins found. So this experiment clearly showed that DNA was transferred from phage to the bacteria, thus establishing its place as the fundamental carrier of the genetic material. So the results and conclusions were like this. Hershey and Chase were able to prove that DNA from the phages is inserted into the bacteria shortly after the virus attaches to the host. Using a high-speed blender, they are able to force the bacteriophage from the bacterial cell, that is called as absorption. The lack of 32 labeled DNA phosphorus remaining in the solution after the bacteriophages had been allowed to absorb to the bacteria showed that the phage were transferred into the bacterial cell. So these were the result and the conclusions of the Hershey-Chase, which concluded that DNA was the genetic material, not the protein. They showed that in growth, protein has the no function while DNA has some function. However, no specific conclusion were made regarding whether material that is sulfur free enters the bacterial cell after phage absorption. What was that? Further research was necessary to conclude that. So bacteriophage is DNA that enter the cell and not a combination of protein and DNA where the protein did not contain any sulfur. So this was given by the Hershey and Chase. But later on we know that there are viruses. Some viruses have the RNA as a genetic material. So what about that viruses? We have seen many scientists prove that DNA is the genetic material of each and every cell. So what about the viruses, other viruses which are having the RNA as a genetic material? So later on some scientists proved that if DNA can act as a genetic material in that cell, the cell which is absence of DNA in that RNA is acting as a genetic material and it is passed from one progeny to its next progeny. And here a plant viruses were selected and that were chosen for the proof of the RNA as a genetic material. So the genome of viruses may be a DNA or RNA. Most of the plant viruses have RNA as their hereditary material. Franklin Conran in 1957 conducted experiment on tobacco mosaic virus, that is TMV, to demonstrate that in some viruses RNA is acting as a genetic material. So before studying about the RNA as a genetic material, first we are going to see about what is TMV, that is the tobacco mosaic virus. TMV is a very small virus composed of a single molecule of spring-like RNA encapsulated in the cylindrical protein coat. Different strains of TMV can be identified on the basis of their difference in the chemical composition, for their protein codes and in differences in symptoms of the tobacco leaves. The tobacco plant which is infected by the virus, that virus is called as the tobacco mosaic virus. By using the appropriate chemical treatment, proteins and RNA of TMV can be separated. In this figure, you can see the TMV structure. The capsomer or the capsid, which you can call, it is made up of protein, and inside that there is the folded nucleic acid, which is the RNA, the genetic material which is the RNA. The capsomer, it is the protein coat and the nucleic material is incorporated inside that. So this is the structure of the TMV virus. Now the discovery of RNA as a viral genetic material. All known cellular organisms have DNA as their genetic material. Some viruses, however, use their RNA instead of DNA. Tobacco mosaic virus is composed of RNA and protein. It contains no DNA. In 1956, Gaius Carmen showed that when they purified RNA from TMV is applied directly to tobacco leaves, they develop mosaic disease. Pre-treating the purified RNA with RNase destroyed its ability to cause the TMV lesions. Means what happened over here? We have seen in the every macleod experiment, they took the RNA also. In that they took the RNase enzyme which destroyed the RNA. Still the colonies or still the growth was seen. But when these scientists, Scarman scientists, took the RNase enzyme and destroyed that RNA, there were no lesions of TMV were found. So which means that RNA is the only genetic material which was passing from one progeny to the next progeny in the TMV virus, because they were having the lack of DNA. So in 1957, Franklin Conrad and Singer showed that in TMV infection with the virus containing RNA from one strain and protein from another, the progeny virus were always the type specified of the RNA, not by their protein. So TMV, this experiment was done in 1956, and after that same experiment was performed by Franklin Conrad and Singer scientists in 1957. They demonstrated that RNA is the genetic material of the TMV. Now you can see in the diagram that Alfred Geyer, Gerhard Scarman, hence Franklin Conrad discoveries of the infectious RNA in the TMV. You can see their images. These were the scientists who proved that the RNA is the genetic material in the TMV. So now proceeding further, we are going to see their experiment. What was their experiment? So in one experiment, protein and RNA were components of the TMV. They were separated and both were used to infect the tobacco leaf separately. It was observed that in case of protein subunits, there was no symptoms on the leaf and no progeny of virus were obtained. So RNA part caused the infection and symptoms appeared on the tobacco mosaic leaf. So fresh progeny of TMV were also obtained. So in other experiment, two strains of TMV, A and B, were shown different symptoms. One causing spots in random pattern and other one in the definite ring form of pattern were taken. So these proteins and RNA parts were separated and the chimera, that is hybrid virus, were synthesized using the RNA of type A and type B and vice versa. So these chimera, that is hybrid viruses, were used to infect the tobacco mosaic virus, and then it was observed that symptom on the leaf always belong to the virus strain from which RNA was taken. Fresh progeny also belong to the same strain. When the hybrid is on the reconstructed viruses were rubbed on the live tobacco leaves, the progeny viruses produced were always found in the phenotypically and genotypically identical to the parental type from where the RNA has been isolated. Now this experiment we are going to see in the diagrammatic way. What was exact experiment given by this scientist? So you can see the TMV particle, they were taken, the infection was seen. When they took the protein, no infection was seen. When they reconstructed the virus, when they took the RNA, the infection was seen, and when they took the whole virus, the infection was also seen. Same you can see in the diagram, Conrad scientists which proved that they took the two strain of the TMV, strain A and strain B. From one strain they took the RNA, and from the second strain also they took the RNA, and the protein were also taken. Now what they did, they mixed that A and B. So when they mixed, you can see that the blue part, which is the protein part, was from the RNA B, and the RNA which is from the A type of species was incorporated. A hybrid virus was made. But hybrid virus, when the hybrid virus was in contact with the tobacco leaf, the infection was seen as the infection was seen in TMV A type of virus, not the B. Both the viruses showed the various type of infection. So the infection which was seen by the hybrid type of infection were the type A of the progeny, because the RNA was of A type. In the TMV-B, you can see the degradation of the protein was made. Here the DNA was of type B and the RNA was of type A. So the protein was of type A and the RNA was of type B. Here also you can see the infection is the type of infection in which the RNA is present. So this experiment concluded that the protein is not responsible for the type of infection. It is the RNA. What type of RNA that virus contains, same type of infection would be there on the leaf. So it is proved that the RNA is the genetic material which is passed from one progeny to the next progeny, and if it is hybrid also, the whatever DNA molecule or whatever RNA molecule is been incorporated inside the cell, that only will have the type of infection on the particular type of plant. So this was explained by the scientist coherent in their experiment. So we have seen our two experiment, that is the Hershey-Chase experiment, that is about the bacteriophage, which use the bacteriophage and prove that the DNA is the only genetic material in the viruses also. We have seen its results, we have seen its conclusion. We have seen the two experiment of the plant viruses, that is the Gerer-Scarman experiment of the tobacco mosaic virus and the continued experiment of the tobacco mosaic virus by the Franklin, Courant and Singer in 1957 and their conclusion. First they show that the RNA is the molecule which is the genetic material of the TMV virus, such type of virus which doesn't have the DNA. In that, RNA is playing a vital role as a genetic material, and in later on the next scientists proved that whatever mixture or hybrid progeny is formed, the nuclear material which is present, the RNA is of which type, such type of only infection it can be caused. So it also proved that the RNA is a functional unit of that particular cell. Whatever type of genetic material is present in the cell, only that type of outcomes will be seen in the microorganisms. So here we have completed our evidences of the genetic material. Now we are going to see the next part in our next lecture. So in next lecture we are going to see the type of nucleic acid, that is the RNAs and the DNAs. We are going to see its structure of DNA, in that nitrogen bases, nucleosides, nucleotides, polynucleotide chain which is having the phosphodiester bond, which is having various bonding. So what bonds are involved in the DNA structure, how the DNA is being made, how the DNA is being incorporated in the cell, and the different forms of DNA. So that we are going to see in our next lecture. So thank you for attending the lecture. If you have any doubt, you can ask me. Thank you.